In this video, we will begin examining gene function. We will discuss the difference between genotype and phenotype, examine the transfer of genetic material, and explore the process of transcription. Cells use their DNA to code for products necessary for cellular structure and metabolism. Gene expression refers to the processes by which the genes are used to produce those products. It is important that you learn the difference between genotype and phenotype. Genotype refers to the genes that an organism contains within its genome. Genomes and genotypes differ. The genome of a cell includes all of the genes and the nucleotide sequences that connect those genes. The genotype is just those nucleotide sequences of the gene that carry the instructions for the products needed by the cell. Phenotype refers to the physical features and functional traits of the organism. The genotype of a cell determines the cell's phenotype because it specifies what types of RNA and proteins are produced. The proteins produced include the structural, regulatory, and enzymatic proteins that determine cell shape and metabolism. While the genotype does not directly code for carbohydrates and lipids, or for specific behaviors, the proteins that are coded for allow those types of molecules and behaviors to be produced by the cell. All of the genes of the genotype do not have to be activated. Some of them may become inactivated permanently or activated only at certain periods of time in the life of the cell. Let's consider two cells in your human body, an epithelial cell and a muscle cell. Examining these two cells with a microscope readily allows us to see that they have quite different phenotypes. If these two cells were taken from the same individual, they would have identical genotypes. Their phenotypic differences are a result of the different genes being inactivated. Gene expression involves two steps, transcription and translation. The genes in DNA are not directly used to produce proteins, but instead are copied into RNA through the process of transcription. The form of RNA that contains the necessary message for the synthesis of proteins is known as messenger RNA. The messenger RNA is then read by the ribosomes to produce proteins through the process of translation. The central dogma of genetics refers to this flow of genetic information, DNA transcribed to RNA, which is translated to protein. Let's examine transcription in more detail. There are several types of RNA that can be transcribed from DNA. RNA primers are forms of RNA produced to allow DNA polymerase to begin to construct new DNA nucleotide sequences during DNA replication. RNA primers were discussed in a previous video. Messenger RNA, also known as mRNA, is the type of RNA that carries the genetic information for the sequence of amino acids from the DNA to the ribosome. Ribosomal RNA, also known as rRNA, is the form of RNA that combines with the proteins to form the ribosomal subunits. Ribosomes, as we have learned, are the organelles responsible for protein synthesis, which is accomplished through the process of translation. Transfer RNA, which is also known as tRNA, are RNA molecules that bind to amino acids in the cytoplasm and then delivers them to the ribosome. The tRNA molecules deliver specific amino acids to the ribosome based on the sequences found in the messenger RNA. Let's move now to the specific steps that occur in the process of transcription. We will examine bacterial transcription first, and will then compare it to that of eukaryotic transcription. Bacterial transcription occurs in the nucleoid region of the cytoplasm of bacteria. Transcription proceeds by three steps: initiation, elongation, and termination. Initiation of transcription occurs when the enzyme RNA polymerase binds to a region of the DNA known as a promoter. Promoters are nucleotide sequences that are located near the beginning of a gene. In bacteria, a protein known as sigma factor assists RNA polymerase in recognizing and binding to the promoter. There are different types of sigma factors and different promoter sequences used by bacterial cells to control the events of transcription. After the RNA polymerase is bound to the promoter, it then begins to unwind and unzip the DNA molecule within the promoter region. The RNA polymerase moves along the DNA, forming a bubble as it moves. Transcription does not begin in the promoter region. Transcription begins approximately 10 nucleotides away from the promoter, where the gene begins. RNA polymerase uses triphosphate ribonucleotides to synthesize and elongate an RNA transcript. Only one of the two DNA strands is used for the process of transcription. RNA polymerase, like DNA polymerase, will only add to the 3' end of a growing chain. Therefore, the new RNA transcript is built in the 5' to 3' direction, and the 3' to 5' strand of DNA serves as the template to be read by the RNA polymerase. RNA polymerase does not require a primer to begin the synthesis of the RNA transcript. As RNA polymerase moves along the DNA template, it matches the incoming ribonucleotides to the template according to complementary base pairing, but instead of using thymine, it incorporates uracil. The RNA polymerase links the adjacent RNA nucleotides together by joining the phosphate of the incoming nucleotide to the 3' hydroxyl group of the nucleotide in the growing transcript. Termination of transcription requires that the RNA transcript and the RNA polymerase that are tightly associated with the DNA template be separated from the DNA strand. Bacterial cells have two mechanisms by which they terminate transcription. The first mechanism by which bacteria terminate transcription is accomplished with a self-termination sequence. Self-termination sequences are composed of two inverted repeats spaced by a few other nucleotides. Inverted repeats are sequences on nucleotides that can loop back on themselves because of their complementary bases. The inverted repeats have a GC-rich region followed by an adenine-rich region. As the polymerase transcribes the GC-rich region, it slows because the number of hydrogen bonds that must be broken makes unwinding the DNA more difficult. The RNA polymerase is slowed down enough that it allows the transcript enough time to start to form hydrogen bonds between its own symmetrical sequences, causing the formation of a hairpin loop. As the RNA polymerase begins to synthesize the adenine-rich region, the hairpin loop creates tension between RNA polymerase and DNA, causing them to separate. The other mechanism employed by bacteria to terminate transcription makes use of a protein known as Rho. The Rho protein binds to the 5' end of the RNA transcript and moves along the transcript toward the 3' end. The Rho protein wedges between RNA polymerase and the DNA template to separate them, ending transcription. The process of transcription in eukaryotes differs in several ways from that seen in bacteria. First, the process occurs in the nucleus of the cell, as well as inside any mitochondria or chloroplasts that are present within the cell. Eukaryotes utilize three different types of RNA polymerase to carry out transcription in the nucleus, and a different RNA polymerase in the mitochondria, whereas bacteria only have one type of RNA polymerase. Eukaryotes do not use sigma factor to aid RNA polymerases to bind to promoters, but instead use several types of protein transcription factors. The final major difference between bacterial and eukaryotic transcription is that the mRNA transcripts must be processed before leaving the nucleus to be utilized for translation. Processing of eukaryotic transcripts involves three events. The first event that occurs is the addition of a modified guanine to the 5' end of the mRNA transcript. This guanine is referred to as a 5' GCAP. This cap is necessary for the ADS ribosomes of eukaryotes to recognize and read the mRNA. The second event that takes place is the addition of adenine nucleotides to the 3' end to form a poly-A tail. Approximately 100 to 250 adenine nucleotides are added to the mRNA transcript. It is believed that the poly-A tail protects the 3' end of the mRNA transcript from damage. Any nucleotides that would be lost would be those of the poly-A tail rather than those important to the message needed to synthesize a protein. The mRNA transcript that contains a 5' GCAP and a poly-A tail is now known as a pre-mRNA. The pre-mRNA contains regions known as exons and introns. Exons are nucleotide sequences that have expressed code for amino acids. Introns, however, are intervening sequences that do not code for amino acids and therefore must be removed from the mRNA transcript. Introns are removed via a process known as splicing. Splicing requires enzymes known as spliceosomes. The spliceosomes cut the introns from the pre-mRNA and connect the remaining introns to produce a functional mRNA molecule that may be used for translation.